And today he's going to talk about how endogenous retroviruses have become an integral component of the host immune response. Let's give a big welcome to Professor Bruce.
Okay, well, thank you very much. It's really a pleasure to be here. I'm going to talk to you quite generally at first about the work we do and what motivates it, why immunology is so interesting to me and why I think it's so important, something about the evolution of the immune system, and then I'll turn to this specific question and tell you how we know that this is the case. Here the red line represents median survival of human beings in different populations. And it may come as no surprise to you that how long we live depends very much upon when we live in human history and also where we live in the world. It might also not be much of a surprise. In the United Kingdom in the year 2000, actuarial studies showed that median survival of human beings was about 80 years, just a little bit less than that. Today it's probably even a couple years longer. In the same year in Mozambique, median survival was about 39 years. And that might not come as too much of a surprise to you either. The fact is that most of the death in Mozambique and elsewhere in the world where there wasn't good development or medical facilities was caused by infection. And you'll notice also that the drop-off in the curve is sharpest between the ages 0 and 5. That's infant mortality, and almost all of it is due to infectious disease. What comes as a great surprise, or did come as a great surprise to me, was that in the city of Liverpool, England, in the year 1860, when England was a great imperial power and controlled much of the world with its navy, in Liverpool at home, median survival was about 10 years. That was what you could expect as a neonate in Liverpool, 50-50 chance of making it past 10 years of age. And again, the main cause was certainly infection. Surprising also is the fact that if you go back earlier in European history to the city of Breslau, 1690, or even to Neolithic times or Paleolithic times, the situation wasn't very much different. One can conclude that the Victorians were living pretty much in the wild state when it came to surviving infectious disease. We don't know really how far back this situation goes, but obviously infection was a tremendous selective pressure on our species, and fully half of us didn't ever make it to reproductive age over most of our history as a species. Even today, if you look across the entire world, you find that about a quarter of all deaths are attributable to infection. That's not the major cause of death. Cardiovascular disease kills more people these days. However, we have to say that there's something rather special about infectious disease death, and that is the fact that it disproportionately affects young people. And because infection affects young people, it's stolen more years of human life than any other cause of death. And as I said just a moment ago, it's the greatest selective pressure that our species has to contend with. Now, we've been aware of microbes only as the cause of infectious disease only for about 150 years. We have to ask, what has this kind of selective pressure done to our species? And what has it done to all species on Earth? Obviously, it brought the immune system into existence. And we would like to look at that a little bit and inquire as to how that came about. It's generally felt, although we don't really have proof of it, that in the earliest situation, when it was microbe against microbe, or microbe against the earliest eukaryotic cells, probably antimicrobial peptides were of key importance. And it's also intuitively obvious that maintaining a system for constant production of antimicrobial peptides is metabolically expensive, and therefore a system for sensing when microbes were present and inducing an immune response would have followed soon after. We know that even today, of course, antibiotics are made by fungi that kill microbes, and we know that there are antimicrobial peptides made by bacteria and fungi as well. So this kind of system for sensing and signaling must have arisen at some point in evolution, and there's good evidence that it predated the split between plants and animals. The evidence for that is the fact that in plants and animals alike, you have very similar protein structures that are involved in sensing and responding to microbes. For example, you have the toll-like receptors in animals. These are these question mark shaped proteins that are single membrane spanning receptors, and they directly engage, in this case, lipopeptides from microbes and recruit adapter proteins to cause a signal. They have a leucine-rich repeat structure. So too in plants. In both rice and also in Arabidopsis, you have cell surface leucine-rich repeat proteins that also recognize molecules of microbial origin. They signal by way of non-RD kinases rather than having a recruitment domain called a TIR domain. But the very same kind of kinase is recruited to the TIR domain indirectly. This is IRAC4. It's homologous to the non-RD kinases of plants. You also have TIR domains in plants in cytoplasmic proteins that are part of a different response system, also leucine-rich repeats there. And one could go on further, but suffice it to say that even though things are scrambled, it's quite clear that early in evolution, these sorts of motifs developed to allow sensing of microbes and delivery of a response. Later, specialized cells developed in multicellular organisms, phagocytic cells that had migratory properties that could wander the interstices of tissues and find microbes and had the potential to kill them. And this was the origin of innate immunity, which persists in many life forms today, including in us, where it's most definitely essential for life. Even if you should happen to lose neutrophils alone, you're going to be very susceptible to infection and you won't survive very long, even if you have an adaptive immune system. Nobody knows exactly when plants and animals diverged. The animal line carried on through dinoflagellates, it's believed, and it proceeded rather slowly in evolution as far as we know. The fossil record is almost non-existent before 550 million years ago or so, because there weren't hard parts of the bodies of multicellular organisms to allow fossilization. But then something absolutely amazing happened over a period of only about 50 million years, and that, of course, was the Cambrian explosion. During that time, there was a tremendous ramification of life forms, and out of the Cambrian explosion came all the life forms that we know today. Animals like this have been preserved in the Burgess shales, and we know that at this time eyes developed, a segmented body plan. We had axial and radial symmetry both. There were teeth, there were limbs. All of the things that we associate with modern life today took origin at this time. And then moving forward, we can see that Cambrian life became the vertebrates and invertebrates with which we're familiar now. There was something really remarkable about vertebrate life, though, and its new take on immunity. The vertebrates are jawed and jawless, and the only surviving lines of jawless vertebrates or agnathans are the lamprey and hagfish, and all of these have jaws. And something remarkable happened at the origin of all of those lines. A new cell was born, and this we call the lymphocyte. It doesn't exist in any of the other lineages of animals, not in insects, mollusks, or elsewhere. And the lymphocyte looks like that. In fact, there are quite a few kinds of lymphoid cell. They all have a nucleus with a smeared appearance of the chromatin, a thin ring of cytosol, and it was these cells that became the substrate for adaptive immunity to develop. They were made in different sites in the body and are even today in the existing vertebrate lines. In the jawless fishes, they were made in an organ called the typhlosole, and as it was later found in the tips of some of the gill structures. Whereas in mammals, they are produced by the thymus and by the bone marrow, in birds by the bursa fabricius. And in all cases, these cells are endowed with immune function. But there's something really remarkable about how adaptive immunity developed. In the common ancestor of jawed and jawless fishes, we know that already there were B-like cells and T-like cells. These would correspond to those cells that come from the bone marrow, those from the thymus. And we know that because we can see surface markers that distinguish these lines, and they exist both in the jawed and jawless products of evolution. The conversion of these cells from what might have been an innate immune function or what might have been some totally different function to adaptive immunity depended on the introduction of a recombinase gene system. And it's thought that that happened by horizontal transfer of genes from microbes. Two genes that have no structural similarity to each other, RAG1 and RAG2, were transferred both to the jawed and jawless vertebrates, and they were able to cut and recombine pieces of DNA, genomic DNA, within these cells so as to allow rearrangement and diversification of receptors. Both the jawed and jawless vertebrates can make trillions of types of receptors capable of recognizing any molecule that they might ever encounter. But what's remarkable is that in the jawless vertebrates, which are so rare today, these receptors are based on leucine-rich repeats and look a good deal like toll-like receptors, and then also exist in a secreted form. These are the binding receptors for antigen in those species, whereas in us, in mammals, in birds, in jawed fish, amphibians, reptiles, here immunoglobulin-type repeats were the substrate for the RAG genes, and we have antibodies that are made and secreted. It's important to remember that the adaptive immune system came along much later than the innate immune system, and it didn't come into a vacuum. It was something that was born in coexistence with a well-developed, functioning host resistance system that was already ancient when it showed up. We know, and it maybe shouldn't be a surprise, that the adaptive immune system is largely dependent on the innate immune system in many ways. If you look even at the response of T cells, we know that they don't respond to antigens that are delivered to them directly. They have to receive the antigen in the context of a major histocompatibility protein, either type 1 or type 2. There are cytokines made by the innate immune system. There are cell surface molecules that are all required to co-activate the adaptive cells to a state where they really begin to respond to antigen. Nonetheless, it's clear that the adaptive immune cells were also born with their own innate immune capabilities and had an ability to acquire even more of them, whether cell autonomous immunity or immune functions that were sort of inborn, we could say. And they were able to do this, of course, because they shared a genome with the whole of a multicellular organism at the time they showed up. At present, we know of 5 major innate immune sensing pathways, and I'll talk about them just briefly. We know about the toll-like receptors, which I showed you examples of a moment ago, and these are those leucine-rich repeat proteins. They're capable of detecting many microbial signature molecules. Probably the TLRs alone are the most ancient sensing pathways and the most capable of detecting all types of infectious organism. We have cytoplasmic RNA helicases that can detect single-stranded RNA, and they work via a single non-redundant protein called Mavs, as we'll talk about later. And these are the RIG-I-like helicases collectively. Then we have cytoplasmic leucine-rich repeat proteins that have either card domains or pyrin domains. These are the NOD-like receptors. They form the inflammasome. And they respond in an unusual way to a huge diversity of insults, we could say, from particulates to microbes to other things, chemical insults of various kinds. And nobody really knows how they do it. It's not as though they're necessarily receptors for anything, but they form the core of a response system that leads to interleukin-1 beta maturation. And that cytokine acts via a protein receptor similar to a toll-like receptor and signals accordingly. There are C-type lectin receptors like dectin-1 or Minkel that detect carbohydrates characteristic of microbes. And then there's a double-stranded DNA receptor recently identified by James Chen at UT Southwestern. This is an enzyme called cGAS that engages double-stranded DNA if it's in the cytosol, recognizes it as something that shouldn't be there, and makes a metabolite called cyclic GMP-AMP, a new second messenger, which in turn binds to STING, a protein in the endoplasmic reticulum, and that signals in a variety of ways. There may be other sensing pathways that are not known yet. Some are minor, some might even turn out to be major, but that's what we know at present. I think it's a mixed audience, so I'll show you a little bit more about how these pathways look. I already showed you a more realistic illustration of toll-like receptors because they've been studied structurally quite extensively now. These are receptors that directly bind microbial products. They recruit adapter proteins that have structural similarity to the TIR domain. In turn, they recruit a layer of kinases, then a ubiquitin ligase, and then a number of transcription factors are activated, either NF-kappa B, AP1, IRF3, IRF7, and hundreds of genes needed for the innate immune response are activated. These receptors exist both on the surface of cells and also in endosomes within cells. And they were touted for a long time as the key bridge between innate and adaptive immunity, but it turns out that they're not essential for activating adaptive immunity at all, at least not where most adjuvants are concerned. The NOD-like receptors are best exemplified, I think, by NLRP3. This is a protein complex in which you have a nucleotide binding domain, a pyrin domain, leucine-rich repeats. It's normally repressed in the cytosol and not very highly expressed. But when the cell is activated, it will polymerize. It will immobilize a protein called ASC, or a second protein called cardinal, and that will activate a protease called caspase-1. Caspase-1 will process the pro form of interleukin-1 beta, making the mature form, which can cause inflammation. And finally, you have the Mavs pathway and the cGAS pathway. These are the key pathways for detecting nucleic acids in the cytoplasm. They work by detecting double-stranded RNA, especially if it has a triphosphate structure at the 5' end. Either short forms or long forms are recognized by RIG-I or MDA5 or LGP2. These proteins then become ubiquitinated by TRIM25, a ubiquitin ligase. They activate a mitochondrially associated protein called Mavs, and it will activate much the same pathways downstream, leading to an inflammatory response at the transcriptional level. The DNA sensor was really hard to find, was sought for a long time, many false stories about what it was. What it appears to be is, as I told you, the enzyme cGAS that makes cGAMP, a nucleotide metabolite that activates STING, and STING also signals, again, much in the same way, although not exactly in the same way. Now, there's been a lot of discussion about the role of the innate immune system in activating an adaptive immune response. And we know today that the toll-like receptors are not required, but they are sufficient, because even in the 1950s, it was known that LPS was an adjuvant. And today we know that it works by way of TLR4. The same is true for lipopeptides and double-strand RNA. They have adjuvant effects. They can signal by way of toll-like receptors. But you knock out all TLR signaling by knocking out MyD88 and TRIF, two of the adapter proteins. You get a perfectly good response to Freund's complete adjuvant, which has mycobacteria in it, to alum, to all kinds of microbes that you might infect the animal with. It's not as though they're needed for an antibody response. In fact, no individual innate response pathway has ever been shown to be essential for an adaptive immune response to begin. And it's quite reasonable to wonder whether any pathway is required or whether this is an entirely redundant system and one pathway might cover for the others. Let's think about what an adaptive immune response is. First of all, I'll tell you that there are two major types. Some adaptive immune responses are dependent on T cells, and others are not, and are called T-independent antibody responses. The T-dependent antibody responses involve protein antigens exclusively. They're degraded, and peptides are presented to T cells bound to major histocompatibility proteins, and that can activate the T cell if co-stimulatory molecules are also expressed. Where T-independent or T-independent or TI humoral responses are concerned, we have two forms. We have the type 1 and type 2 humoral responses. And let's talk about those for just a second. The type 1 T-independent response is initiated by toll-like receptors directly on B cells. There's no T cell involved here. And that will lead the B cell to become activated and to make an effector plasma cell or a memory cell. LPS will drive this, for example, and it's believed that this is all activation mediated by toll-like receptors. This is how one gets antibodies to LPS, which are not proteins, to lipopolysaccharides. In the type 2 T-independent B cell response, the activation is often mediated by a polysaccharide, sometimes by a virus that has a capsid with a repeating protein structure. But what appears to be needed is very extensive aggregation of the B cell receptors on the surface of B cells. No toll-like receptor input is required, and one again gets plasma cells that secrete antibody and memory cells that can go on later to become plasma cells under this stimulus. So antigen alone is sufficient, provided it's capable of extensively cross-linking the BCR. And nonetheless, we wondered whether there might be any other requirements for the type 2 T-independent antigen response. And we happen to have in our extensive collection of mutations derived from forward genetic screening things that affected all of the known innate immune pathways. And we thought, why not look at all of the types of humoral response we can and see whether any of these innate immune pathways are required. So this work was done in my laboratory by Ming Zhang, and these were the mutants which I've color-coded for you here that he decided to test. He had a central mutant of the RIG-I-like helicase pathway, mutants affecting the cGAS, cGAMP, STING pathway. All of these mutants, which affect TLR signaling, either individual TLRs or in some cases all TLRs. He had one mutation within the NLRP3 pathway, and then he had an IκB kinase gamma mutation which affected several pathways. We didn't bother to look at the rather at the C-type lectin receptors, but we did look at 4 of the pathways. he took all of these mutants and wild-type mice as a control, and he immunized them with NP-ficol, nitrophenylated ficol, a polysaccharide-type ligand that can drive an adaptive immune response that's T-independent, a type 2 T-independent response. And he looked for IgM that was specific for NP, for the hapten nitrophenol. And here is the response of the wild-type mice. You see that most of these mutations really had no significant effect on the antibody response, but a mutation in STING did have an effect that was significant. In CGAS, an even somewhat greater effect. In Mavs, about the same. And very clearly, NF-kappa B activation was important because an I-kappa B kinase gamma mutation almost completely abolished the response. One of the first things we wonder about when we see such a response is this really B-cell intrinsic? And we were able to look at that very easily by taking RAG-deficient mice that have very few T or B cells and infusing B cells from each of the mutants into them, or from wild-type mice as a control, and then doing the immunization. And we found, indeed, this was a B-intrinsic problem. If you had mutations in STING, Mavs, or CGAS, you had a very diminished response to antigen challenge. Now we know that the type 2 T-independent response depends largely on two types of B cells. Marginal zone B cells, which are in an anatomic location in the spleen called the marginal zone, and B1 B cells, which are peritoneal B cells that don't arise from the bone marrow as most B cells do, as B2 cells, which are the majority do. Instead, they arise somewhere from an extramedullary source and renew throughout life. And originally, both of these types of cells arise from the fetal liver rather than the bone marrow, at least in large part. We wanted to know whether there might be a developmental problem with these cells and whether they might be scarce in those mutants. And so we looked quite carefully by flow cytometry at different classes of B cells in the spleen and in the peritoneal cavity. And we examined wild-type, STING, CGAS, or Mavs mutants. To make a long story short, mutations in these genes really didn't affect the fraction of any of these types of B cells. So there's no developmental problem to easily explain what's going on. However, the mutations in STING and CGAS and Mavs do impair antigen-specific B cell activation and proliferation. And we can judge that by either immunizing or not immunizing, as you see from the bars, and looking here at the spleen marginal zone B cells, or at two subsets of the B1 cells, B1A from the peritoneum, or B1B, or at peritoneal B2 cells. And we find that with immunization, we get simply to have more of the antigen-responsive NP-positive cells, and therefore clearly there's a response defect caused by errors in these pathways. Now it's really curious to us. We know these pathways are important for RNA and DNA sensing, and the mystery began to arise in our minds, where is the RNA and DNA coming from? Here we're injecting the mice with NP-ficol. There's no RNA or DNA being given in the injection. Where does it arise? Of course, it could come from some exogenous source. Maybe we were killing tissues by doing these injections, although we doubted it. And nonetheless, we began with a hypothesis, as shown here, that there must be cytoplasmic DNA and RNA to activate these pathways. There would have to be an interaction with the antigen receptor. And apparently, by blocking CGAS or STING or Mavs, at least in part from each of the two pathways, we had a problem with B cell activation and proliferation. So we set about to investigate where the DNA and RNA were coming from. It is probably known to some of you that in your genomic DNA, something like 45% of all sequence is ultimately traceable to retroviral origin. And in fact, well-conserved or reasonably conserved endogenous retroviruses make up something like 8% of our genomes. Not all of them are inducible or expressed. Some of them have become degenerate and have become so-called line repeats that lack the LTRs. Others sign repeats, which are parasites that live off of line repeats. But these seem to us to be a likely source of the cytoplasmic RNA and DNA. The idea being that when such an endogenous retrovirus is expressed, it first makes an RNA copy. That RNA copy could be detected by Mavs, or by the Mavs pathway, by Rigi-like helicase, let's say, and then by Mavs. Or it could be reverse transcribed and become a double-stranded DNA molecule and be sensed by C-gas. And that was the model that we began with, that somehow extensive cross-linking of the B-cell receptor might lead to activation of endogenous retroviruses. We would get retroviral RNA. It could either then be directly detected by Rigi, MDA5, and Mavs and lead to a signal for sustained B-cell proliferation, or it could be reverse transcribed, be detected by C-gas, then by the second messenger C-GAMP activate STING, and that would give signals for prolonged B-cell activation. If we immunize mice with NP-ficol, then we can demonstrate that indeed endogenous retroviral RNAs and DNAs are elevated in antigen-specific B cells as compared to non-antigen-specific B cells. This is a very nice experiment because it's internally controlled. One can identify those B cells that have an antigen receptor that will recognize nitrophenyl by using a special stain that contains that group. And one can look selectively then at those cells, separate them into different populations by cell sorting, and then look at the amount of RNA or DNA encoding the retroviruses. And one finds with different classes of retroviruses and different members of the class that not always, but in most cases, there is a significant increase in retroviral RNA or in reverse transcribed DNA if you do sequencing on these pools. And often it's by more than a log, and in no case is it in the wrong direction. So evidently, retroviruses are induced simply by immunization in vivo under the conditions we normally use. This is the flow cytometry profile. I doubt you can see it very well, but the cells that are reactive with NP are quite rare, but they're definable and detectable. Not only will the transcript be induced, but also proteins encoded by retroviruses are induced. And we looked at two such proteins, first at reverse transcriptase activity in the NP-positive cell population compared to the NP-negative, which was not really different than the naive cells from mice that weren't immunized. We also used an antibody that was very broad in specificity. It recognized an epitope of the envelope glycoprotein common to all murine leukemia viruses. And so we were looking only at one class of retroviruses. But here we have immunized B6 mice in the first two lines and then non-immunized B6 mice at the bottom. If you immunize and you separate the NP-positive fraction, there you see there's a shift in expression of that protein. So the B cells make active retroviruses that are at least capable of making their encoded proteins, which is not to say infectious particles are secreted, but no question immunization causes the activation of these ERVs. It was our opinion that probably the Rigi-like helicase family of proteins would sense the RNA. And we decided to see whether that was so. We used an RNA immunoprecipitation assay In this assay, one takes a lysate from cells, an antibody against a tag or against a protein of interest. In our case, we were interested in Rig-I-like helicase. And we immunoprecipitate the RNA, at which point we can do various analyses. In our case, we did reverse transcription and sequenced the RNA species. And we compared, as a control, the precipitation of glyceraldehyde phosphate dehydrogenase, which we wouldn't have reason to think would be an RNA binding protein with that kind of specificity. So here we're looking again at NP positive B cells and NP negative B cells, and it should be clear to you that the presence of Rig-I precipitated target endogenous retrovirus is higher in the NP positive cells than in the NP negative cells for all of these different classes of ERV. James Chen collaborated with us throughout this project, and one of the, well, let's say one of the requirements of the hypothesis would be that when you activate these B cells, there is something about their RNA that's inflammatory in an assay system that involves Mavs as the central detector. He was able to take RNA from NP positive and NP negative cells that we prepared and to transfect it into a target reporter line, whereupon he could measure interferon beta produced in excess by RNA from the NP positive cells, or CXCL10, a chemokine ligand, or interleukin-1 beta. All of these are inflammatory proteins, and all of them seem to be driven to expression by something in the RNA pool from those cells. Going a little bit further, James was able to show also that that RNA that drove expression of those targets was dependent on Mavs. So you can perhaps read, if you're close up, that this is a BLAC6 recipient cell. This is a Mavs, rather BLAC6 donor cell or a Mavs deficient cell. And Mavs is required to drive the response. And we see this for all of these cytokines once again, and for several other cytokines as well. Now, of course, to this point, we've only been looking at individual mutations in Mavs, in Sting, in CGAS, and we began to think about combining them because it appears that these pathways are partially redundant. You can signal the presence of cytoplasmic RNA through this pathway, cytoplasmic RNA alone, or it can be reverse transcribed and go through the CGAS-CGAMP pathway. And we decided first to look with a pharmacologic inhibitor, with a reverse transcriptase inhibitor. In BLAC6 mice, you see a reverse transcriptase inhibitor will very weakly mitigate the response, the antibody response. In a Sting deficient mouse, it has really no effect that one can see. And in a Mavs deficient mouse, it has a very dramatic effect. So all these are low to begin with because they have a Mavs or a Sting mutation. But if you block both pathways, then you're in fairly serious trouble where the T-independent response is concerned. So there we have blocked with reverse transcriptase inhibitor that pathway. We've blocked this pathway with a Mavs mutation. We wind up with almost nothing. Also, of course, one can combine the mutations genetically, and this we did by using a CRISPR approach, taking a CGAS knockout, making a Mavs knockout within it. And as opposed to the single knockouts, which have the partial impairment, again, a bit more than the Sting mutation, the double knockout is close to zero in its response. There is a little bit left, suggesting there's some other residual pathway for the type 2 T-independent response, but it isn't all that much. Now, is this only applicable to NP ficol? That's something pretty exotic. Those of us who are getting up in our years are encouraged to have an immunization with Pneumovax at least once to protect us against pneumococcal pneumonia. This is in a vaccine in which simply a number of serotypes of pneumococci are pooled, and their capsular polysaccharide is extracted and given as an injection. And we found exactly the same thing. The CGAS Mavs double mutation would prevent the response to Pneumovax, just as was the case with NP-Ficol. So here we have our working model again. I've shown it to you before. We believe that both retroviral RNA and DNA are upregulated. cGAMP must be produced in order to activate STING, and this is another measurable endpoint that we had to look at. That can best be done by mass spectrometry. And what one can see in the case of B cells that are NP positive, taken after immunization, is a blip of CGAS, which has its exact predicted molecular weight. And from NP negative B cells, you don't see that, nor do you see it in naive B cells, whether they're NP positive or negative. We've done a time course and found that peak levels are seen at about 4 days, but significantly high levels of CGAS are present in antigen receptor positive B cells even out to 10 days, indicating that this is a very prolonged signal, probably what's required for differentiation to form plasma cells. We know, too, that cGAMP has an immunostimulatory effect on B cells, both in vitro and in vivo. And while it won't completely rescue a cGAS mutant by bypassing it, it will at least work in that direction. And this may have something to do with other effects that we've observed more recently, that it causes apoptotic effects when administered from outside the cells. We're not really entirely sure, but that's consistent with the general idea that we have in mind. Other requirements of this model are that, first of all, the response be dependent on the B cell receptor. Also, it's known that a protein kinase called BTK is critical for type 2 T-independent B cell responses, and that should be part of the picture. And we also would like to think that there's NF-kappa B dependency in this model. If we take mice deficient in BTK, indeed, then they don't make an NP-specific IgM response. We can also look with an antibody against the B cell receptor, and we find that that's blunted by a BTK deficiency as well. We know that NF-kappa B dependent cytokines and interferon beta are upregulated in the NP positive but not NP negative B cells after NP-Ficol immunization. And here we're looking at TNF, IL-6, IL-12, interferon beta, all hallmarks of the response. And we see that they're selectively elevated when the cell is positive for the NP recognizing antibody. So this is consistent with the idea, as all of these are NF-kappa B dependent to one degree or another, that NF-kappa B must be activated. But we have also in our collection this viable hypomorph of I-kappa B kinase gamma. And normally the knockout is lethal, but this has a rather serious deficiency of I-kappa B kinase gamma, yet it's a living mouse. Here, if we go back to the old experiment and we look at all the different classes of retroviruses, we find that they're simply not induced. And so it seems to be that NF-kappa B is really the principal driver there, as the model suggests. Retroviral transcription is induced by anti-IgM, and it's dependent on intact I-kappa B kinase gamma. And we can see that again with a number of different classes of retrovirus, and with anti-IgM antibody administered. In the open bars, you see you don't get a response if you have I-kappa B kinase gamma deficiency. This is an important experiment because it had been suggested that maybe complement activation, for example, might also be a critical route to activating the B cell, but it seems not to be the case because while NP-ficol might activate complement, simply activating the BCR with an anti-IgM antibody wouldn't be expected to do that. There is some complexity here because not only the B cell receptor, but also these pathways that we're talking about are capable of activating NF-kappa B. Mavs can do so. It might be thought also that CGAS activation can do so, and you come to a kind of chicken and egg problem and wonder about which is really the most important pathway involved. We looked at that, taking Mavs deficient and CGAS deficient mice and using an anti-IgM antibody as the activator. And of note, we found that in Mavs deficient cells, if we look at NF-kappa B P65 translocation to the nucleus, or if we look at P105 phosphorylation, the Mavs deficiency really has a significant effect. It diminishes activation, whereas in CGAS deficiency, there's no significant difference from the wild type, suggesting that Mavs does probably feed back and sustain the response by activating NF-kappa B as well. And we also find that NF-kappa B translocation in anti-IgM activated cells is affected, and differentially so. So here the order is reversed. This is the Mavs deficient cells, and you see the Mavs deficient cells have less translocation of NF-kappa B to the nucleus as compared with the CGAS deficient, where there's no significant difference from wild type. When all is said and done, we feel quite confident in the model I'm showing you now, that you have cross-linking of antigen receptors by polymeric antigens, be they polysaccharides or viruses, for example, with a repeated capsid structure. This activates BTK. BTK leads to the activation of NF-kappa B. NF-kappa B drives endogenous retroviral gene expression, and one has both viral protein synthesis and, to a degree, reverse transcription of the product. And reverse transcribed DNA with retroviral sequences is recognized by CGAS, which makes this new metabolite, the cyclic GMP-AMP, and that activates STING. On the other hand, the RNA can directly activate RIG-I and Mavs. Mavs will activate NF-kappa B, helping to sustain B cell activation and proliferation. STING, on the other hand, will do other things. cGAMP definitely is active for a long time in the cell, and STING would be activated to make other transcription activators, and together they contribute to a robust IgM response within just a few days. Now, this leads to some summary conclusions and a few inferences. First of all, we see that two innate immune response pathways, Mavs and CGAS, have both been harnessed for a new and special purpose in B cells. This is not to say it doesn't happen elsewhere. It might happen elsewhere as well. But at least in B cells, these formerly outward-looking receptors that evolved to detect pathogen RNA or DNA seem to detect RNA and DNA that ultimately originates within the B cell itself. So they've become somehow at the core of the adaptive immune response, where once they were innate immune proteins, and they function in a strictly cell-autonomous manner. We've come to think of retroviruses, including endogenous retroviruses, as bad. And obviously they are bad and can do bad things to us. But it has to be said in their defense that at the same time, they're needed, almost absolutely needed, for the type 2 T-independent B cell response. And that's an important response. I should emphasize that. It's the response that most protects us from things like pneumococcal pneumonia, and probably from many viruses as well. We can say that as soon as a retroviral element becomes part of the germline, obviously it's subject to positive selection like all other parts of the germline, and we should look at it in that context. One other way to look at it is that here a retrovirus has been co-opted to fight against other kinds of pathogens. That is something that we have seen on a very specific scale with viral interference among different types of retrovirus, for example, but we've never seen it in this sort of a broad context where retroviruses in general, perhaps thousands of them, cooperate to combat completely different types of pathogens, encapsulated viruses and encapsulated bacteria. We have heard, and we know that it's correct, that there are autoimmune diseases caused as a result of a failure to degrade the products of retroviruses, especially retroviral DNA. There's a famous instance of an enzyme, an exonuclease called TREX1, which, when mutated, well, let's put it more elegantly, when TREX1 is mutated, an autoimmune disease results known as chilblain lupus or Aicardi-Gutierrez syndrome. We've come to think of this in a way as an exaggeration of the physiologic response of B cells, in the sense that expression of endogenous retroviruses is something physiologic. We know that now. It contributes to the normal antibody response. One thing that I most wonder about is whether retrotransposition sometimes occurs in B cells once they've been activated by an antigen. This, of course, could have mutagenic effects. And it's also possible that even if in black 6 mice there isn't secretion of retroviral particles, in some of us there might be. I'm not counseling you not to get your Pneumovax shot and not to be immunized, because I think that most certainly outweighs the bad effects. But we do have to wonder if the normal immune response, the adaptive response, sometimes leads to retroviral production, and that might lead in turn to retrotransposition or insertion into other cells in the neighborhood, and whether that might have neoplastic effects. So could this be a new link between inflammation and cancer? Most people would point to things like oxidative radicals as being of key importance, or simply de novo viral infection or other kinds of mutagenesis that might go on. But I would say that possibly this is a link, and it's certainly something that we will look at more in the future. This is my group as it stands now, and out of all these people, there are 4 who work on the screens related to adaptive immunity, and only one of those 4, Ming Zhang, was the one who did all of the experiments that I showed you. We had extensive help from James Chen, who is in the Department of Biochemistry at Southwestern, and from Xiaodong Li, who's a postdoctoral associate in the Chen lab. Also from Ralph Deberardinis' lab, and Zeping Hu and Xiaolei Shi in that lab. And we've collaborated steadily with Gunilla Carlson-Hedestam and Jerry McInerney on this overall project as well. Thanks very much for your attention.